Good evening, everyone. Thank you once again for joining us for this, our first Sunset series that is hosted by the NNIA. We are very, very grateful to have you joining us today, and hopefully we are going to have a hassle-free session without any glitches in power and all of those things that are part of our daily lives lately. We were running a poll on this side of the world around where the funnest places that we have joined webinars from. We're not going to share, but I think we've all joined webinars from all sorts of interesting places in the past couple of years. But yes, well, let's get started. Without taking up too much time, just a reminder as per usual. Let me see if I can get my slides up. We are the Nestlé Nutrition Institute, and as you know, everything that we do is about the science of nutrition, and we do not in any way promote the use of any product. So by joining us this evening, we do not expect you to be recommending any of our Nestlé products. So starting us off today, it is going to be Jonas Hauser. I hope I got that right. Anna Marie said, I just have to remember the Jonas brothers. So there we are. He's joining us from Switzerland, and he will be presenting to us. My slides are not very clear. I don't know if my team can support me on that. Are you able to assist with the screen? A little bit more? All right, no, it's fine. We are going, so Jonas is going to be taking us through HMOs and their role in brain development. As we know, HMOs are everybody's favorite at the moment. All healthcare professionals are all about HMOs because of their link to the gut microbiota. And we know that the gut microbiota is responsible for so many things. It's associated with many conditions, and brain development is one such, and we are proud to share what it is exactly the research at Nestlé Research has uncovered to date. Soon after Jonas's presentation, we will quickly go into Nobe's one. Nobet, he is not Nobe, but he is Nobet, and he will be taking us through human milk oligosaccharides, translating scientific discoveries to pediatric practice. And I already have a few questions for him on this very, very topical subject. So we just want to remind you, if you are new to the Institute, you haven't joined us before, we encourage you to use your phone. If you're joining us from a laptop, just scan the QR code here, and then the QR code will take you directly through to our registration page, and then you'll be able to get first-hand information on this and future activities. So Jonas, you're up first. Thank you very much.
Good day. I'm Dr. Jonas Hauser, and today I will give you a presentation on the physiological role of human milk oligosaccharides, which I will abbreviate in HMOs, with a specific focus on brain development. First, I want to give you a very simplified overview of brain development that we can split between the functional and the structural aspect. On the functional aspect, we know that the brain developed from early in the pregnancy. And especially, we know during the first year of life, there is a first wave of functional development, which is the sensory motor function. Indeed, as pediatricians or as parents, we all know that newborns have the capacity to move around and see or hear their environment mostly. And then in the second half of the first year, we see the emergence of language with some noises and whistle and little words or beginning of words that are being pronounced. And then following this first year of life, we see the development of progressively more complicated functions such as memory, planning, decision-making in later adolescence, for example. And that's for the functional aspect. On the structural aspect, we know there are like sequences of events that are partially overlapping that starts as early as shortly after the conception, for the neurulation, for example, and continue with initially mostly the neuronal population being prepared either in proliferation or migration, and then the connectivity of the brain being set up with the synaptogenesis and the myelination. And what is really interesting for us and for the general brain development is that while some of the processes are prenatal, others are going postnatally up to adulthood, such as myelination, for example. Now, if we think about what can modulate the brain development, there are multiple modulators that exist. I give some example here. So we know, for example, the microbiome composition can have an impact on the brain development. We know also, of course, stimulation and parenting during early age has an impact. We know nutrition has an impact. The social, cultural, as well as the environment influence have an impact. And finally, but not the least, genetic and biology, of course, has an impact. In this talk, I'll focus on one aspect of nutrition. And when we talk about nutrition in mammals, we talk about breast milk. And just a side note to acknowledge the magnificence of breast milk is that it's an evolution that took about 300 million years in making. And it started from the ancestors of mammals that were laying eggs with a parchment-like shell that was intolerant to desiccation, and therefore the evolution developed glandular skin secretion for moisture. And from this protection role against desiccation and hydration role, the evolution moved towards this glandular skin secretion into a nutrient-rich milk that take the role to vehicle nutrients and energy to the newborn. So we move from protection to nutrition. As I mentioned, in mammals, we're talking about breast milk. In breast milk, we have multiple components. First of all, we have water that is providing hydration. Then we have nutritive components, for example, lactose, proteins, and fats. These are mainly digested, and they have nutritive value. They contribute to healthy growth and development. And then we have some bioactive components, for example, the HMOs. These are not necessarily digested. They don't have a nutritive value. They can support or be used by the microbiota. And these have a support for brain development and immune function. And I will focus on the brain development in the presentation. So now let's focus really on these human milk oligosaccharides, these HMOs. As I mentioned, they're part of the solid component of the milk. And in general, they are between 5 and 15 grams per liter. And this is the third component of the milk in terms of solid component after lactose and lipids. And if we look specifically at HMOs, we can see there's like more than 200 different structures that have been identified, of which about 20 are making the bulk of it. And as you can see here, but I'll go a bit more in detail on the next slide, we can split them into core structure, fucosyl HMO, or silyl HMO. And this is the molecule, the structure of the HMOs. As you can see, it's a glucose linked to a galactose, and then you have additional sugar that are linked to that. And depending on which sugar are integrated, we can cluster them in different families. If they contain a fucose, we call them fucosylated HMOs. If they contain a sialic acid, we call them silylated HMOs. And if they contain neither, we call them neutral HMOs. And you can see that the structure can be elongated and become more complex with different sugars. Now, what is interesting is that HMOs stand for human milk oligosaccharides, but we find them in other mammals. And here I just want to make the specific comparison of breast milk, so in human, to bovine milk and formula milk, because formula milk being mostly produced from bovine milk, it's logical to show the bovine milk. So just a repetition, breast milk, about 5 to 15 grams per liter of HMO, and quite a huge variety in terms of structure. If we look now at bovine milk or infant formula, we can see the amount is much lower, between 0.05 to 0.2 grams per liter of oligosaccharides, and the diversity is extremely limited. We have now more and more infant formula, including some specific HMOs, but we never reached the complexity of the breast milk of more than 200 different HMO. So high HMO levels and diversity in breast milk, low HMO level and diversity in formula and bovine milk. Now, what are the functions of the HMO? So we know they have an impact on microbiome development. They can be acting as prebiotic. They also have an antimicrobial effect. We know they have an impact on the gut barrier function. We know they support the immune competence development, but what I will focus and what is my main interest is the modulation of the development of the nervous system as well as of cognition. So on the right end of the axis here. And I'll go now through the different evidence that have been published so far, linking specific HMO to brain development. And then I'll go a bit through the mechanism of action and come with the conclusion. So the first study published was by Berger in 2020, and this was a landmark in the HMO knowledge because it's the first clinical data. And they've used an American Hispanic mother-infant cohort of 50 subjects. They collected the breast milk at 1 and 6 months and analyzed it for HMO. And they measured the neurodevelopment using the Bayley-3 scale, which is a neurodevelopmental assessment tool. And what they've reported here on the right-hand side is a clear positive association between the level of 2-fucosyl-lactose in breast milk at 1 month of age and the infant cognitive development measured using the Bayley-3. And as you can see, it's a positive slope. So the higher the level of 2-fucosyl-lactose, the better the cognitive development of the infant. This was in the same year, there was a study in Malawi mother-infant pairs, 659 subjects from 0 to 2 years of age. They measured the breast milk HMO concentration at 6 months, and they focused on the communication development inventory using the MacArthur and Bates tool. And what was interesting in that study is instead of focusing on a specific HMO, they said, let's measure the fucosylated HMO or the silated HMO. And then they split the population with population receiving milk with high or low fucosylated HMO and high or low silated HMO. And what was really interesting is that they've shown that in the population with high fucosylated HMO, at 18 months of age, the language development was increased in terms of number of words known by the infant. And you can see this was significant, and similar findings were obtained for the silated HMOs. So these are showing that the family of HMOs are playing a role. In 2021, Oliveros and colleagues published another paper linking specific HMO to cognitive development in infants. This was in a Spanish mother-infant pairs, 81 subjects. The breast milk was analyzed at 1 month of age, and the Bayley-3 scale was used at 6 and 18 months. And they reported two main findings: a positive association again between 2-fucosyl-lactose at 1 month of age in mother milk and composite motor scores, so the motor development at 6 months of age. And second association was between the 6SL, 6-silyl-lactose level in mother's milk at 1 month of age, and the composite cognitive score at 18 months of age. And if you think back to the development of the function across age, it's quite logical that you would see a more beneficial impact on motor development at 6 months of age, because that's an age where the motor development is peaking, whereas the cognitive development, if you think at 6 months old infants, is relatively limited. On the contrary, at 18 months, you know that the cognitive development is starting to peak, and therefore you expect to see a potential impact there. In 2021, we published in a collaboration with the University of North Carolina a study leveraging the Brain Connectome project, an NIH project, in which we brought the Nestle nutrition knowledge. And what we've identified here is that in an American mother-infant pairs, 99 subjects, we had a sequential recruitment of the subjects, so we age regressed statistically the age from the data. So we measured the breast milk HMO concentration between 0 and 2 years of age, and we used the Mullen scale, which is a similar tool as the Bayley to measure neurodevelopment. And what we observed is that we segregated the population in subjects with A-tetra positive breast milk, so with A-tetra HMO in the breast milk. And we saw a general composite score of the Mullen scale association with 3SL. However, what we also saw is that this association was driven almost entirely by the expressive language. So again, we've linked 3-silyl-lactose with language development. More recent study done in Brazil, where Brazilian mother-infant pairs, about 70 subjects followed from 1 month of age to 12 months of age, made the association between level of HMO in breast milk at 1 month and used the age and stage questionnaire, social emotional one. This was looking specifically at potential inadequate personal emotional development during the first year of age. And as you can see, they had a huge list of HMO on the x-axis, on the y-axis, sorry, and on the x-axis is the different domain in which there could be problem. And what was interesting is after the correction for multiple comparison, LNT, so lacto-N tetraose, which is a neutral HMO, was associated with a reduction of risk of personal social or more than 2 domain problem in more than 2 domain. So this is the first study that is extending the potential functional benefit of HMO beyond fucosylated or silated one to the neutral one. And last year, Berger and colleagues published a follow-up study in an Hispanic American mother-infant pairs, 20 subjects. And the number is quite low, but this is because they perform an MRI study. So they use magnetic resonance imaging at 1 month of age, and they correlated the finding with the breast milk level of 2 fucosylactose, 3 fucosylactose, 3 silylactose, and 6 silylactose. The study has a huge number of outcomes in terms of MRI, but I'll focus on one, the fractional anisotropy, that is a measure of the connectivity a bit of the brain. And what is really interesting here is that and I take just 2 fucosylactose and 3 fucosylactose as an example, they are 2 fucosylated HMO, so in the same family. But very interestingly, if you look at the heat map, it's showing the impact on fractional anisotropy. And you can see that 2 fucosylactose has a negative association with fractional anisotropy. On the other hand, 3 fucosylactose has a positive association. And this is the heat map on the top, and they show specific correlation graph with the regression line for one of the brain area, the inferior parietal cortex for 2FL and the anterior corona radiata for 3FL. And you can see that these are opposite directions. So one is a negative correlation, the other is a positive correlation. And I think this is giving a very interesting insight on the potential differential role of specific HMO, even within one family of HMO. Now, one question that I've been asked to me multiple times is, okay, you've shown us that HMO are having an impact on brain development. What is impacting the HMO levels? And there have been multiple studies looking at that. I do a very succinct summary of that here. So the first point is that has been highlighted since a long time is the genotype. We know that mothers can be FUT2 positive or negative, or FUT3 positive or negative. And this is called the secretor status and the Lewis positive negative status. And you can see on the right-hand side, if you use secretor, so it's the group 1 and group 3, you have 2FL. If you're non-secretor, group 2 and group 4, you don't have 2FL. And this was the first finding. And then the Lewis type has also been identified as impacting on the composition of the HMO. And you can see indeed, if you're Lewis positive, the group 1 and group 2, or Lewis negative, group 2 and group 3 and group 4, you can see there is differences. What is interesting is with these 2 genotype changes, you have a complete change of the pattern of HMO present in the human milk. So that's one aspect. And when you look at the overall pattern of human milk oligosaccharides in the breast milk, you can see here non-secretor on the left, secretor on the right. The main difference is the big blue bar missing on the left. So this is the absence of 2FL. But you can see also that other fucosylated HMO are impacted, like LNFP1 in light violet is reduced in non-secretor. LNFP2 on the other side, just below it, is increased in the non-secretor. What is also interesting is that you have impact on other HMO, such as the silylated, the 3SL, 3-silylactose in yellow, which is decreased, and the LNT, lacto-N tetraose, which is in dark blue, which is increased. So the overall pattern of HMO is changing. Another study did an interesting effort in taking multiple environmental variables or maternal variables such as age, lactation stage, parity, season, study site, and try to do the association of these variables with specific HMO. And what was interesting in that study is, of course, lactation stage, this was known, popped out as a modulator of the HMO level. We know some HMO are stable across lactation, some increase and then decrease, some decrease from the beginning. So this is quite known. The parity was also showing some impact on one of the HMO. But for me, what was really surprising is actually the season of birth was impacting the level of HMO. So this is, and I put that here because I think it's important to keep in mind that sometimes some variables that for us seems not really relevant actually are relevant. And the last one, which for me is extremely interesting, is that specific nutrient intake have been associated with specific HMO. This was a nice matrix on the right-hand side where you can see, for example, I highlighted in red 2-fucosylactose, 3-sialactose, 3-FL, and different nutrients at the bottom like fructose or glucose, as well as polyphenol. And you can see they have a significant impact on the HMO level in the breast milk. Now, one slide where I'll just go and give an overview of the three most likely mode of action of HMOs. So the first one, which is pretty evident, is the impact on the microbiota. And this can be by having HMO modulating specifically the microbiota composition, as well as having the microbiota metabolites changing depending on the presence or absence of HMO and the interaction between HMO and microbiota. The second one, which is coming more from a preclinical studies, is the modulation of the vagal tone by fucosylated and silated HMOs. And this has been demonstrated in the preclinical models where the effect of 2-fucosylactose, the beneficial effect of 2-fucosylactose on neurodevelopment was dependent on an intact vagus nerve. So using a vagotomy, researchers have demonstrated that you lose part of the effect of 2-FL when you have a vagotomy. Whether 2-FL itself acts on the vagus nerve or via the microbiota and its metabolite, this hasn't been answered yet. And the last one is a potential modulation of gene expression at the level of the brain. And this we've demonstrated again in a preclinical studies where we used genetically modified animals to evaluate the impact of milk with 6-sialactose versus milk without 6-sialactose. And what was incredibly interesting for me is that when we look at the gene expression in the brain, we saw in a spatial, so in brain region specific, and in a temporal specific aspect, a modulation of several pathways related to neurodevelopment, general neurodevelopment pathway, as well as myelination pathway. So it has a clear, the presence or the absence of 6-SL has a clear impact on this neurodevelopment, and this can be supported by the potential role of 6-sialactose as a carrier of salic acid, and we know salic acid is important for myelination, for example. So these are the three main mechanisms of action that have been proposed by different researchers and that are currently being further investigated. Now, I want to give you some take-home messages that if it's the only thing you need to remember about this presentation, they are here. The first one is that early life is a key window of opportunity to modulate brain development. And this is not solely through nutrition, but through all the modulation that I've presented and other I haven't talked about. The second is that nutrition is one of these influencing factors of early brain development. And this is especially true during the first 2000 days. And we focus now on the 2000 days. Before we were focusing on the 1000 days, and maybe tomorrow we'll focus on the first 3000 days. What is important to remember is more that nutrition plays a role from preconception, because the stores that will be available for the fetal development are built in preconception stage, through conception and in early life, up to adolescence at least, if not adulthood. Then I've shown you the existing evidences linking specific human milk oligosaccharides in terms of their intake in early life and the cognitive functions or the brain structural development. And I think the sum of the evidence are accumulating over the years and are showing that there is a clear role there. I also highlighted that there are multiple maternal factors that can modulate the breast milk level of specific HMOs, and that diet is one of them. I've highlighted the three main mechanisms that are hypothesized to mediate the HMO effect on neurodevelopment, and these are the microbiotal composition and metabolites, the vagal tone, and the brain gene expression. And I want to thank you very much for your attention, and I'm looking forward to your question.
Hello, everybody. It's a great pleasure for me to be here today with you and talk about the human milk oligosaccharide from research to pediatric practice. My name is Norbert Sprenger, and this is my conflict of interest. I'm an employee of the Société des Produits Nestlé in Switzerland, Lausanne. Now let's start looking at the context of the human milk oligosaccharides. And you all know that in early life, you start to be colonized in your gut with the gut microbiome. So this colonization is actually a very dynamic process in the first couple of years of life. And that has a major impact on the immune system. You know that the gut is obviously our primary organ to digest and absorb nutrients. But besides that, it's also our major organ for immune development and for immune education and for immunity in general, because it's such a big surface. And the colonization with microbes that is illustrated here on the left side is actually a driving force for this immune maturation. And now there's a lot of interactions happening through the gut that goes to long-term health, and this not only for immunity, but also now more and more recognized for immunity in the lung, but also for other body organs that are modulated somehow by microbial metabolites through the gut. And you can just see here and on the illustration of how we describe today the microbiome, there's a kind of a dynamic maturation process that can be described as a trajectory that goes diagonal to about 2 years of life, and then it kind of levels off to a certain constant microbiome that stays as we have it then later as adults. And especially in early life, it is extremely malleable. And that is why it's so important to have the right nutrients and the right feeding for the microbes to develop in the right appropriate way. Now, just a few words also on the immune system. So you know that the immune system is composed of many, many factors, and I just break them down here as the different layers. So the first layer is a resilient gut microbiome. So this is our first layer, and this you all know that it's a very important layer of defense, because when you have antibiotic treatment, you're getting often an antibiotic associated diarrhea because of the lack of this defense layer of the gut microbiome that is brought in. Then we have the epithelial barrier function. So the epithelial cells are the next layer of defense. So you know that this is highly glycosylated with a mucus layer, but also on the cell surface. There's lots of glycans, and these glycans are all resembling actually the so-called human milk oligosaccharides, because they're made by the same machinery. And then we have passive immunity. So this is immunity brought in by the mother, for example, through maternal secretory IgAs. And then slowly, as the infant matures, you get other immune factors kicking in. For example, natural innate immunity with the secretory IgA produced in the gut of the infant that starts at birth and then gives you a long-term also defense layer. And then obviously the long-term adaptive immunity that comes in with exposure to antigens, with vaccination and so on, that you learn how to tolerate food, for example, but also how you learn how to actually defend yourself against pathogens and viruses. So see that these are the different layers, and these are the different layers that we try to address with nutritional interventions and with nutritional solutions to help have the infant develop these in an appropriate way, because this has then long-term consequences overall in the infant. Now, when we talk microbiome, we should also talk about the metabolites that the microbes produce. This is considered their language. So that's how they communicate to us, to the body of the infant or to the adults. And you can appreciate here on this slide a couple of organic acids with the short-chain fatty acids acetate, propionate, and butyrate. And you can see also the organic acid lactate, just as examples. And what you can see here over the first 2 years of life, that you see a first coming up of acetate that is followed by propionate that comes in a little bit later. While lactate is also very early in a peak when you have lactic acid bacteria like the bifidobacteria and then lactobacilli. And then later, when you start introducing complementary diet, you get more and more of the so-called butyrate-producing bacteria, and then you get the metabolite butyrate. So you can see that there is a sequence of events. Not everything happens at the same time. And this is an important aspect to keep in mind, because we want to also have an age-appropriate modulation of the microbiome so that you have the best health outcomes. Because probably there's an education process early in life that is different from the next window of opportunity in the second age when you start introducing solid food compared to breast milk. And then this is an important aspect, because the communication between microbes and the infant can have an effect on the immune system, but also on later metabolic health and obesity, and more recognized to have a big impact also on the brain development and temperament. So this is important to keep in mind, and to keep in mind that we have an age-appropriate maturation of the metabolic signature and of the language that is coming from the microbes to the body of the infant. Now, this is also an illustration of the importance of the gut microbiome. And when we look at different maternal factors or birth factors, we can recognize many that have an impact on the gut microbiome development of the infant. So first of all, the maternal diet. So this is not so much a direct impact on the gut microbiome of the infant, but it has, through the metabolites that the mother produces in her microbiome, an impact on the immune system development in utero of the growing infant in the mother. And then gestational age, this has a very big impact, and this you all know from preterm birth. So with preterm birth, you have a lot of problems and a lot of risk factors for many diseases. And here the microbiome is actually very different from a normal term-born infant. Then mode of delivery with C-section birth, that is an impact on the microbiome. And that impact has later consequences also for health outcomes, like, for example, allergies or infections. Then antibiotic exposure, this is the most studied and obviously the most evident that it has an impact on the gut microbiome. And here I just give you on the right side a few examples of how antibiotic use in early life can have an impact on immune competence development, but also on metabolic health, obesity risk, and then also more and more recognized on cognition and temperament, which is another topic of our research and of the discussions that we have today. Now, I mentioned, I had illustrated on the other slide, that diet has also a big impact on the gut microbiome. And in the early diet, you have breastfeeding. And in breast milk, you have the so-called human milk oligosaccharides. So this is our main topic of today here in this presentation. And the human milk oligosaccharides are shown in different basic research models and in observational trials, and now also in intervention trials, to have different impacts on the gut microbiome by, on the one hand, deflecting pathogens, that's illustrated here. On the other hand, boosting beneficial microbes like the bifidobacteria or certain specific bifidobacteria. And this can have a consequence then on the immune development based on the barrier function here, but also with metabolites and maybe with some HMO that goes systemic on the direct immune cell interaction and development of immune system and immune competence, but also, and this is likely again through metabolites, on the nervous system in the gut, but also in the central nervous system with effects on cognitive development and temperament. So this is all here summarized, and it's in a recent review that I'm happy to share with the team if needed. Now, today we talk about 5 HMOs very specifically. And what I try to do in this slide, which is a little bit busy, is to summarize what we know on the 5 HMO, specifically on the different functions that are today described. So let's start on top here with the microbiome maturation. So here we have different names. We have 2'-fucosyl-lactose or DIFL, LDFT, LNT, LNNT. So these are all primarily described to have an effect on gut microbiome maturation. Then you have another set of HMOs, again with 2FL, LNT, but now also the CN-related ones with 3SL and 6SL that may help immune competence development through different mechanisms. And these mechanisms can be direct interaction with certain receptors or again through the maturation of the gut microbiome. And then you have interactions that are reported here again for a different set of HMOs with 2FL again included, but also 3SL, 6SL that have data that show that there is an effect on gut and brain communication and cognitive development. Now, this shows you that you have some specific functions, but also some overlap on the functions. So why is there this overlap? Maybe part of it is the gut microbiome. Another part may be just redundancy in the milk that you can ensure that the infant development goes appropriately by having redundant functions in the different molecules that are delivered by the milk. And now let's see in the next couple of slides just a few examples of what we learned when we do observations in breastfed infants and we try to associate to the breast milk HMO composition and different clinical outcomes. So this is just illustration of the different papers that are out there on infections. So there's quite a lot. This was the early data that came in that showed effects, especially on diarrhea prevention, but then also now on respiratory and I would say more and more actually going towards the respiratory infection risk reduction. Now I show you an example here of a study that we had in a cohort in Dhaka in Bangladesh. And here you can see on the left side the incidence per 100 infant years on the respiratory infections, and you see there's a peak and then kind of it goes away more or less. And during this peak on the early infection, acute respiratory infections, we see a protective effect of certain breast milk components that are fucosylated in a 2' position that are triggered by the so-called secretor gene or the FUT2 gene. So here we can see this significant effect in the 0 to 6 months, which we lose then with time. So during the exclusive breastfeeding period, we seem to have a protective effect on respiratory infection based on some of the HMOs that are in the majority actually of the mothers. Now, when we come to allergies, the situation is slightly more complicated because not all of these studies show you a positive effect. Some of them do, and a positive effect in terms of association, but there is some indication that in certain conditions, HMO may have a role to play in allergy prevention. And I show you an example here from our own study. And here what we could do is we see in the C-section born infants that we have an alleviation of the bifidogenic effects through breast milk that is not given when you have the so-called 2' fucosyl HMOs missing. That's shown here, that these are the furthest away from the vaginal born infants in blue. The red one here are the C-section born that miss the 2' fucosylated HMOs. And when you have 2' fucosylated HMOs in these breast milk samples, then you can see the infant gut microbiome actually moves closer to the vaginal born infants. And this actually correlated very nicely to the observation that we had in the C-section born infants, that the presence of the 2' fucosyl HMOs alleviated the risk of allergies and actually had a later onset of allergies in these infants. So there is a kind of a link maybe through the bifidobacteria. And this also fits actually very nicely with a systematic review that I cite here on the beginning, that early life bifidobacteria were proposed to be a key factor to help prevent the allergy risk later in life. So there's something driven maybe through specific bifidobacteria. Now, partly this data on allergy prevention are backed up by preclinical data. So I show you here an example for 3SL on the left side. This is 3SL here, where there is a help in terms of allergic sensitization in this animal model. And you can see here with more and more 3SL, you basically remove the number of mast cells and the allergic symptoms. Now on the right side, there's another model. This is a kind of a food allergy model, again in an animal model. And here you can see 2 HMOs that helped to basically reduce the symptoms in the allergy, and that's shown here and also here with the 2FL and with 6SL. So we don't know exactly all the mechanisms yet, and we don't have yet clinical data, but this indicates that something happens with certain HMOs on immune competence development. Now let's come to the most interesting part of today, and this is what did we learn so far on the 5-HMO blend infant feeding with a randomized controlled trial. So we have available now these different HMOs, and these HMOs can be tested in a clinical setting by doing a randomized controlled trial. So here we will get really causal interactions, while on the observations we can only get hypotheses, but we do not have causality. So just a little bit of background. What did we observe so far? And this was observations that we had already with 2 HMOs that I show you here. And this was actually a reduction that we had observed on lower respiratory tract infection, and specifically on bronchitis, where you can see that here the relative risk is very strongly reduced and significantly reduced on both terms. So LRTI actually contains also the bronchitis term. It just includes also other terms that are in the lower respiratory tract. And then on the upper respiratory tract, we had another study in cow milk protein allergic infants. So here we see overall a reduction in the number of episodes on respiratory infections, and we see also a trend here on the lower respiratory tract infections. So we kind of repeated the findings that we had here, even so it did not reach significance in this second study. So there is an indication that something through the HMO feeding can help the lower respiratory tract infection risk. Now, what we try to do is to understand where does this come from? What are the signals that are linked to the reduction in the risk of lower respiratory tract? And here what we did is to do a case control study out of the data that we collected. So we now basically ignore the formula feeding and which group they were, and we just compare cases and controls for LRTI that happens after 3 months of age. And these are the signals that we see in the gut microbiome. So on the one hand, we see the signal that higher acetate was on the protective side. So with higher acetate, we had less risk basically for LRTI, while in the cases, those who got LRTI later, they have lower acetate per total short-chain fatty acids. So you see that link to the first slide that I showed you on some of the metabolites, on how that could have an impact later in life. And then what we also found is that we have specific bifidobacteria groups that were actually higher in abundance here in the controls compared to the LRTI cases. And these were the longum group. And then specifically in the longum group, we see this longum subspecies infantis. So very specific bifidobacteria that are known to use HMOs for their metabolic activity. And these were actually linked here to the protective effect that we see. Now, this is backed up by preclinical data that we see acetate helping also from diarrhea, protecting from diarrhea from specific E. coli pathogens, and then also high fiber diet related to higher acetate, and that protected in a mouse model from respiratory syncytial virus, a typical cause of bronchitis in infants. And then last but not least, there's also a link of acetate shown in preclinical models on the production of specific immunoglobulins in the mouse. So there is maybe some particular aspect to be observed in the next evaluations that we do and studies. Now let's come to the 5-HMO trial, and I show you here just the general setup of the trial. So we have the purpose of the trial, and this was to actually make sure that the infants grow well, grow normally, age appropriately, and that the HMOs that we provide in the formula are well tolerated and safe. Now, this is the basically primary outcome. And then we also looked at infections. I don't have the data yet for that, but we also started to explore gut health and immunity and microbiome composition and function. to link actually of what we had observed previously to this new trial, so that we can better understand mode of action of these HMOs now in an infant setting. And this is basically the objectives reporting below, but I want to show you here also a little bit more on the study design. So we had a reference group in orange. These were the fully breastfed infants, fully breastfed to 4 months. Then they were allowed to introduce solid food. And then basically we followed them also up to 15 months to have them as a reference. Then we have formula-fed infants. So those who chose not to be breastfed could be enrolled in the trial within the first 3 weeks of life. And when they were enrolled in the trial, they were randomized to the control formula or to the test formula with 1.5 gram of HMOs per liter, or to the test formula with 2.5 gram per liter. And what I show you primarily is here then the data that we had from 3 and 6 months stool samples, but I also want to show you just as a first set the growth data that we observed and the digestive tolerance and anthropometry data basically. Now, this is most important, obviously, that the formula is safe and well tolerated, and this is given for the formula. So we had absolutely no indication that there was any risk in terms of digestive comfort or any adverse events that would go in a signal that there was a safety issue. What you see also is the growth curves match very, very closely to the WHO growth curves. So this is the reference here in the middle, and that all the 3 formula groups basically grew equivalently. So whether we give the HMOs or not did not affect here the growth of the infants in the first 12 months of life that I show you here. And that continued actually to 15 months. It was just not presented yet. And here this was presented here in a poster abstract. So the formula with the 5 HMO is safe and well tolerated, but let's see what we learned about the gut microbiome. And on the gut microbiome side, I'll just give you here a snapshot on the overall gut microbiome. So this is illustrated here with a so-called diversity plot, where we look at diversity at baseline. You can see those who choose to be formula fed, they were already formula fed at baseline, and then they were enrolled in the trial. So their microbiome was already different from the ones who chose to be breastfed. So this was here the orange one. And then as we go along the first 6 months, you can see that the 2 blue ones, so the formulas with the HMOs, move closer and closer and basically overlap at 6 months with the gut microbiome overall composition of the breastfed infants. So we got closer and we got actually almost identical here to what we have in the breastfed infants. Now, what is the driving force for this change? And this is maybe not surprising now that you can see here the bifidobacteria in this green color that start to increase. So here is the control formula fed at baseline, then the HMO formula at baseline, and the HMO also number 2 at baseline. And you see they're identical in the number of bifidobacteria present in the stool. And then you can see that there is a higher increase here in the bifidobacteria in the HMO formula fed, similar to what is observed in the breastfed. And this is illustrated here with more detail on all the subjects that you see here at baseline. All the formula fed are actually equal, and the breastfed were already higher. And then with time, 3 months to 6 months, we see that with the HMO feeding, we increase the bifidobacteria species abundance overall. Now, maybe more importantly, what we observed is that we have infant-type bifidobacteria species. So this contains groups of species that are particularly found in infants, like the infantis, like the longum, the bifidum, and the breve group. And these are all increased together when we give the HMOs. And then again, maybe a link to what we observed previously. What we see particularly increasing is the Bifidobacterium infantis as a specific HMO utilizing species that increases with the HMO feeding. So indicating that maybe we can expect also some particular functional aspects that we had previously observed. And here I just want to intersect a little bit with a publication that came out recently that showed the importance of HMO utilizing bifidobacteria in the early life immune development. And this is just illustrated here. When you have a high HMO utilization capacity, then basically here there was an observation of much less intestinal low-grade inflammation compared to the low abundance of HMO utilizing bifidobacteria. And then you have different setting with a kind of a pro-inflammatory environment in the gut immune system. And so I wanted to give this citation here, which says that it's not simply the presence of bifidobacteria that is responsible for the immune effect, but it's the metabolic partnership between the bacteria and the HMOs. And this is a very particular combination that you obviously expect from breastfed infants and that we don't have normally in a formula because they wouldn't have the HMOs. Now, what else did we observe in gut microbiome composition? So one important aspect here is the Clostridioides difficile. So this is actually toxigenic one. So Clostridioides difficile that can lead to antibiotic-associated diarrhea. And here what we observed is that the prevalence already and the relative abundance of this toxigenic bacteria is much lower or absent in breastfed infants. And then here at baseline, we have actually more or less equal relative abundance of this formula fed groups. But then at 3 months, we already see a significant reduction here of the Clostridioides difficile. And also at 6 months, we see still a significant lower amount here with the HMO feeding. So we kind of reduce a risk factor through a pathogenic bacterium. Now, what else did we see? And I think here we come to the aspect of the gut ecology and the metabolites that I mentioned initially. So this can be looked at already as a global measure by looking at the fecal pH. And the fecal pH is an indicator of the general metabolic activity of the gut microbiome. And what you see here at 3 months and at 6 months is that we have significantly reduced fecal pH. So more acidification in the colonic compartment of the gut in the HMO-fed groups, the blue groups, and this was similar to what we observed here in the breastfed reference group. Now, maybe this you would have expected already of what I said before. We actually increased the relative abundance here of the acetic acid at 3 and at 6 months, while we reduced the relative abundance of butyrate at the 3 and 6 months. So you remember, butyrate should come up after 6 months in the normal situation. So this we would then follow up and see how we actually increase also the butyrate later. And at the same time here also with the HMO feeding, we see lactic acid increased and propionic acid also decreased. Again, propionic acid should come up at around 6 to 9 months in the time schedule from what we know from the literature. Now, let's look at some gut markers. So one gut marker, secretory IgA, is a typical immune marker that leads to immune defense. So you know that there is passive immunity given by the breast milk with secretory IgA, and that is why we measure here a very high amount also in the breastfed group. So this breastfed group, IgA, is coming primarily from breast milk. So we cannot really compare it to the formula-fed group, which is not having any secretory IgA, because also the infant makes very, very low amounts at birth and then starts to increase. And what you can see here and appreciate is that we have a significantly increase of secretory IgA formation in the gut of the infant. So this is about a doubling. And this is still there at the 6 months time point. So primarily here at 3 months time point that increased compared to the control formula fed. At the same time, we see another marker decreasing with time, and that is here the alpha-1 antitrypsin. Again, it probably is confounded here in the breastfed arm by the alpha-1 antitrypsin from breast milk. But clearly what we see here in the formula fed is a reduction. And this is a marker that indicates the gut barrier function. So there's probably a nice effect also on the gut barrier function that we could observe. Now, what's the take-home message of all what I said until now? So HMOs added to infant formula are generally well tolerated and safe, and they show promising results in terms of supporting the immune competence development. And I think this is largely driven through the help of the age-appropriate development of the gut microbiome with very specific bifidobacteria and their activity. Then the 5-HMO blend was clinically shown to support age-appropriate growth and to be well tolerated. It was shown to help intestinal immune system development and gut barrier function development. And then last but not least, again, I want to mention here the age-appropriate gut microbiome maturation that includes the enhanced growth of specific bifidobacteria that can utilize HMOs and the reduced abundance of toxigenic Clostridioides difficile as the main outcomes until now. So with that, I close here and I thank you very much for your attention. Thank you very much.
Good. Well, welcome everybody again from my side. I'm Annemarie de Beer and also responsible for the NNIA here in Africa and more specifically in the Eastern and Southern Africa region. And I really have the privilege of asking all the questions tonight. So if there are questions that you haven't asked and that you would like to ask, please put it in the chat box. It's quite important. We have a number of questions. And we're going to go through them actually as quick as we possibly can. And Norbert, we will start with you. Norbert, we will start with you. We understood this afternoon that Norbert is actually German, Swiss German, and Jonas, with his English name, is actually French. So we are working this all out still, but Norbert, there was a very interesting question, and I'll start with you. It's asking, are there any data on preterm babies if they have a different brain development in terms of HMOs and the microbiome? And then if they could not be breastfed earlier due to their development, are they able to catch up when they get more quality nutrition?
Thank you, and hello everybody for this nice Q&A session now. I think this is a very intriguing question and it has many elements. So maybe let me start with the first. I think we know that when you can provide breast milk to a preterm baby, you will get the best quality nutrition to the infant. Obviously, the infant, due to its prematurity, cannot suckle from the breast. So often, you have to give it in other ways, and you have to have milk banks often because not all mothers can provide the milk. But I think there you get already, a huge amount of benefits just by providing the breast milk. And this has different elements. So one of them may be the HMOs also, because they will help the gut microbiome to eventually catch up. So now there is still the birth mode and all the antibiotics that you often get as a preterm born baby that will hinder the microbiome to develop in the right way initially. But at least you can provide, the best nutrients through the breast milk to basically catch up on that. Now, specifically on brain, I think there is very little known. So we had actually done one cohort study where we looked at the HMO that the infants got by measuring actually exposure of the HMOs. And we could identify some specific HMO that are also linked to immune development that had an apparent association to a cognitive outcome later in life. So now that's a starting point where we could, start hypothesizing what could be behind, as Dr. Jonas also showed us, and say, okay, can we make a mechanistic sense out of this and basically understand better what is happening? So this was a relatively larger molecule on a fucosylated molecule that we identified there. And other studies usually show effects of HMOs, specifically on the incidence of necrotizing enterocolitis. They associate that. That does not mean there's a causal interaction, but there's at least an association shown. And in terms of the microbiome, I think often you would have to give a probiotic, to basically help because of that high amount of antibiotics that the preterm babies get. But I am a researcher, not a doctor, so you would probably have even more insight on this than me. And thanks for this question. Very good. Oh, yeah, I see that Anne-Marie is frozen. Yeah.
Good evening. I can just step in for a minute while we get Anne-Marie back online. So thank you for that, responding to that question. We do have another question on the chat, and the question is, what is the source of HMO that are added to formula feeds?
So I'm happy to take this question. So, I mean, thank you, Jonas. So obviously we do not isolate it from breast milk because otherwise we would have a much more complex mixture of HMOs already available, but we have to manufacture them by biotechnology. So basically in a fermentation biotechnology process, in fermenters, we produce the individual HMOs, and then they are actually purified to very, very high purity. And once they're highly purified and characterized from all the angles that you could do on the analytical side, they underwent also a safety toxicity testing similar to any pharma compound that we would have on the market. So they go through the preclinical testing with in vitro models on genotoxicity and all the OECD regulated suggested pathways, how to test, and then also juvenile animal models to make sure that we are also in the group of age that we address with the HMOs in an addition to a supplement or a formula. Welcome back, Anne-Marie.
And I'm muted 2 years later. Jonas, we've got a question for you. I don't think it's been asked yet. And this relates to HMO levels. And the question was, does mothers' hereditary, the genes or the race impact on HMO composition and or levels?
So yes, there is plenty of evidence, and actually that's one of the first maternal factor that has been identified to impact on the HMOs in the breast milk. It was the genotype of the mother regarding two fucosyltransferase genes, the FUT2 and the FUT3. And these have been shown to whether you have a single nucleotide polymorphism on them that is making them inactive or active, it will lead to a different pattern of HMOs in the breast milk. There is a recent study that had investigated more in depth other genes that could be impacting on the HMO levels and on the HMO pattern in the breast milk. And they have identified multiple genes, but this is still in the bioarchive, so it hasn't been peer reviewed. But it is showing that it's basically a complex multigenic factor that is impacting. And this is quite logical when you think we have more than 100 different HMOs that have been identified. It is very likely that there's multiple genes that are impacting the levels of each of them. I don't know if Norbert wants to.
No,
Yeah.
Norbert, anything you
Yeah,
want to
maybe-
add?
Maybe just a small addition. I think it's clear that from the effect size, the genetic background on the secretor gene, so the FUT2 and the Lewis gene, the FUT3, are the key factors. So these really change the profile quite dramatically. And then we have to also take into account that different populations, so different heredity races, have different proportions of those genotypes in the population. So this is usually a kind of a confounder if you would not separate by the what we call milk groups by the different genotypes when you analyze. Now, we had some observations that we think even when you are secretor positive or FUT2 positive, that you have some small changes that maybe in some Asian populations, there's a little less expression of the 2' fucosylated molecules. But these are still very, very small population size that we analyzed, and so we cannot have a good conclusion. So it's definitely, much, much smaller effect size than the genotype yes/no analysis.
Good. Then I think there was a very interesting comment that you made, Jonas, around the season of birth. And the question is, so which season is best? I think as health professionals, we can now kind of guide our moms as to when to have their babies. When will be then the best season to have your baby in?
So I'm not a pediatrician, so I won't take the pretension to be able to give an advice on that. I think I was more raising that as an interesting point to indicate that there are probably multiple environmental factors that are impacting the HMOs, and we are at this interesting period of the science development where we start to identify some of them. And one aspect that Norbert raised also is that the amount of the variability that is explained by the factor can be very, very important. We know the genetic is impacting, has a very high impact, and the other factors have usually a lower impact by themselves. But ideally, if we would understand all the factors with their impact, then we could give a recommendation. But as such, I don't think there's a preference of season for birth of a baby.
Maybe I would want to give one addition, because this was an interesting study that Jonas mentioned there. And it's actually one that was shown in an African population, that there was a seasonal effect seen. And the authors themselves, they speculate that it has to do maybe with the diet, because the season comes with different diet. And so it's not so much the season, it's maybe the richness of the diet. But now this is only a hypothesis that they brought in, and we and others started to analyze in different cohorts of how maternal diet would actually have an impact on the HMO composition. Any signal that we saw was extremely weak, and in terms of effect size, a lot confounded by other factors. And I think we don't have a very clear picture yet, unless maybe you're malnourished or really having a problem on malnutrition, that then maybe there is different effects. And this was shown, for example, on cellulated HMOs, that they were lower when you're in malnourished situation.
Good. And I think within this malnutrition world we're looking at, we also see the overweight and obesity as part of the malnutrition. So not just underweight, but also then overweight and obesity, which I expect would then also have some sort of impact, even though we don't know yet.
Indeed, we had seen that with analysis that we did when we look at the pre-pregnancy BMI, and that we had some association with higher pre-pregnancy BMI and some of the, again, cell-related components. Now, effect size was very small, even so we reached significance for some of them. We don't know yet how to interpret it. One hypothesis that we have is that the high BMI mothers have also kind of a slight pro-inflammatory status in their physiology. And this is known to have higher cellulation in the body. So many acute phase proteins are highly cellulated. So maybe the whole machinery of cellulation is kind of turned on. And this, as a consequence, leads to maybe a little bit more of cellulated HMOs also in the breast milk. I mean, that's what we hypothesize. Now, whether that has an impact on the infant is doubtable because of the small effect size.
Good. Well, I think it's just fascinating. We have another question. Jonas, this question is for you. Until when does the beneficial effects or the beneficial cognitive effects last with an early HMO intake? So do moms have to breastfeed for 7 years, or what's going to be best?
So there is actually two questions behind this question. So first of all is like, how long is the HMO exposure that is leading to an impact? And in many of the studies, the population were either European or American. So usually breastfeeding, exclusive breastfeeding, is limited to 2 to 4 months, depending on the most of the time on the law around the maternity leave, which is driving the duration of breastfeeding in many countries. But there are still some people that can breastfeed for a longer period. So this is one aspect, the duration of breastfeeding. And while on the HMO, I don't think there is a clear answer on the duration of breastfeeding, hence the duration of exposure to the HMO and the beneficial effect on the cognition. It is known that the composition of the breast milk is impacting on the cognition, and this has been reported for HMO levels measured at 1 month of age and not at 6 months of age. So it seems that the early phase of lactation is really key, at least in one study that was reported. And there's a very recent paper that came out literally this week or last week, where they looked at a 3-year follow-up of infants that had their HMO measured during the first 3 months of life. And there they show also a beneficial effect for some of the HMO on cognitive function that are developing that late in the development. So the beneficial of HMO might, like the fact that you're exposed to HMO might be beneficial up to longer term. And the duration of the HMO, I mean, this is, I had no opinion before going into the field. Now my opinion is the breastfeeding duration is basically driving a lot of benefits, and potentially HMO is one of the mediator of this. You're muted, Anne-Marie.
Apologies, I don't know, someone muted me, it wasn't me. Norbert, this question is for you. How do ingested HMOs connect to the observed benefits in specific organs like the lung or in the brain?
Yeah, so this is a very good question, because there is actually a lot of debate of what happens actually to these HMOs. I mean, how would they have an effect in a place where they never reach? And you shouldn't get the HMOs into your lower lung, at least, maybe in the upper respiratory, I mean, in the upper tract, in the mouth and so on, to the ears. You could imagine some direct contact. But what we learn more and more is, especially for the lung, that we have a so-called gut-lung axis, and that we can modulate, and I showed you one example with the acetate in animal models, that we can modulate through a modification of your gut ecology and the different metabolites, how the mouse in the mouse model, or even how the infant probably reacts to a pathogen, and how that mucosal immune system is mirrored into other organs. Now, very similar maybe, and this is at least my favorite theory, is that we have also connection to the brain that comes through that gut-brain axis through metabolites, because we know that many of these metabolites that are coming from the microbial host interaction can travel in the body and can have an impact. So many of them can be anti-inflammatory, so there could be always a kind of a dampening of a pro-inflammatory stimulus that could have also an influence on brain development. Or there can be other molecules that stimulate somehow nerve cells, glial cells, and so on, that would have an impact on brain. The other theory that is long time discussed is that some of the HMOs may be helping as a building block, and Jonas mentioned it a bit. I think today we come away from that theory, and we have more data with stable isotope labeling and so on that indicate that this may not necessarily be the case. So that this building block theory is maybe not right, and that there's some other mechanism that moves into the brain. I mean, the vagal nerve is another pathway that is a kind of a highway to the brain even. And so there are maybe these kind of pathways that would be favored. But in my view, there's a lot going on through that interaction with microbes, and the right window, the right microbes, and that right dynamic. And this is, I think, indicated more and more in the literature. And Jonas just mentioned the example of the very early association with 2FL in early life that has the long-term effect, but later it didn't associate. So that indicates, again, we have some windows of opportunity, maybe also for brain development, how we can affect this. And that's why it's so important to get the right starting point. And that's why it's so important to get breast milk, you know.
Correct. Good. We have one last question, and this is now really the last one. If there are more questions, I'm going to ask the audience, please keep on sending them. We will make sure it gets answered in due course, but please don't stop asking questions. The very last question, Norbert, is for you. Is there any link, and maybe Jonas, you can also answer this, is there any link, direct or indirect, of HMOs to hypersensitivity immune reactions, such as atopy and or susceptibility to infants' colds and flu?
I'm happy to do the start. So I showed you some link that we had through the observations and further exploration from our intervention trials with HMO, where we try to basically get mechanism of action from the analysis that we did on metabolites, on the microbiome. And there, I think, yes, there is a link to the respiratory tract infection risk. We don't know if it's exactly flu or if it's RSV or if it's another pathogen, because we did not measure that. So we have only a starting point now. But we see actually a series of metabolites. So it's not only acetate, but we had identified also some sphingolipids and some other lipid metabolites that shouldn't be too high in the first 3 months, because the higher they are, the more we saw actually them in the risk side. And this is also confirmed by a Japanese study that looked at the case control for bronchiolitis, where there was at the same age group, the link made with those sphingolipid metabolites. And then we see others like acetylated molecules or gamma-glutamylated molecules that can travel and that can be taken up from the gut into the body. That seems to be linked also to a protective effect. So I think there's a lot of indirect effects through the HMO feeding, maybe through those specific bifidobacteria that I mentioned, and then maybe other bacterial groups that link to the at least to the infection risk. Now, in terms of atopy, we are far behind. So I showed you just that one example on the atopy risk onset in infants who have a hereditary risk for atopy because one of their parents is atopic. And there we see this delay, or basically this change in the bifidobacteria population. But we see the effect that with the breast milk composition only in the C-section born infants. And there we see also that link to the bifidobacteria. And now there is a second systematic review that came out. I'm sorry, I didn't put it in the slide. And that shows also that there is an early bifidobacteria that seems to be important to prevent the atopy, at least as a consensus. But in terms of mechanism, we don't know yet. Now, I think the HMO have a role there because we know that these specific bifidobacteria are actually HMO users and they can profit from them and make the right metabolites. Sorry, I cannot give you a conclusive answer, but
no, but I
that's
think,
our research
well, we know
field
there's a
here.
link of some sort. Jonas,
Yes.
maybe the last word from you?
No, yeah, I think Norbert explained it very well, and I think it's basically pointing at this very challenging question of trying to understand the interaction between more than 100 HMOs and the microbiota, which is like incredibly complex, and the multiple developmental outcomes in infants, and that's like a pretty challenging tasks that us and several others are trying to answer by asking specific questions. So that's why it's very exciting, because we're at the moment where we're trying to unravel all of that, and every new studies is really like shining a little bit of light on this very complex puzzle that hopefully one day we'll understand.
Thank you. Well, I'm going to have to stop it here. I can hear the excitement in our speakers' voices about doing more research. And I really hope we have instigated or at least ignited some fire tonight, or some flames at least, to do even more research in South Africa and in the ESA region for all our contexts. Yes, and then we've got a thumbs up for that one. I'm going to close the session now, but before I do so, I think there's a few thank yous that is due, and very specifically to our two speakers, both Norbert and Jonas. It was brilliant. Thank you very, very much. I think the fact that this is kind of the latest information is just so exciting. And we really want to thank you very, very much for making time for this. And I must be honest, this is not the last see of you. I think we're definitely going to have a few more of these discussions with our audience that we also have online tonight. Then I also want to just thank Lindiwe I call her Lindy because it's just quicker, but she's actually Lindiwe Wati. And Lindy, thank you very, very much for bringing all of this together. Lindy works very hard behind the scenes to get our speakers and get everything done, get the CPD accreditation going. So thank you very, very much for all your hard work, as well as our team, the technical team, Grant, Vanessa from the source. Thank you very, very much. And then I think last, but definitely not least, I would like to thank the audience that has joined us tonight for the first webinar of 2023. Please keep your eyes open. There's a couple of things. One, we have recorded this session, so it will still be available on this link for a month, after which it will be available on the NNIA website. And then we will make a list, and I haven't asked our speakers yet, but I'm asking them now to make a reference list available for you on the website, whereby you can just see which articles were used, and you can go and look for them, and if you can't find them, we will help you. But I think I think the interest is there, and we would very much like to make it available to all of you. So to our audience and everybody else, thank you very much for joining. Thank you for staying, and I'm sure nobody has to drive anywhere. I hope not, but I just want to say thank you very, very much, and stay safe and have a good evening. Thank you.